First of all, thank you very much to the IMP, the committee, of course, the Max Birnstiel Foundation, for such amazing recognition. I'm truly honored and grateful. I would like to share with you a bit of my PhD journey, where I was particularly interested in understanding which physiological signals are indeed crucial for driving cardiomyocyte maturation. Because, as you may wonder, cardiomyocytes undergo an extensive maturative process from embryogenesis to adulthood, finally leading to the generation of these highly specialized contractile cells that can't stop working a single day of our lives. Thus, it is not surprising at all that cardiomyocytes are highly flexible metabolic cells, able to consume a wide variety of substrates just to be sure that ATP generation never stops. And for me, a very interesting scenario illustrating this metabolic flexibility happens in mice right after birth. Because during embryogenesis, these cardiomyocytes will be mainly consuming glucose as major energy source. However, once mice are born, they need to shift their metabolic profile, stop eating glucose, and now fatty acid oxidation through the mitochondrial compartment will constitute the major pathway to produce energy. And despite this process is extremely important, not only for cardiac function, but also for organism survival, it is poorly understood. And in Mercedes Ricote's lab, back in the Spanish National Center of Cardiovascular Research, we are very fascinated by understanding how cardiomyocytes orchestrate their homeostasis from the transcriptional standpoint. And to do so, we focus on studying a very particular transcription factor named retinoid receptors, which are indeed ligand-dependent transcription factors, meaning that they need to sense different metabolites and molecules in the cellular environment, and in response to them, they will be able to modulate specific genetic programs in the cell. And turns out that the role of RXR in cardiomyocyte was not known. At the beginning of my PhD, I aimed to understand how these transcriptional mechanisms, mainly driven by RXR, were indeed driving and being involved in cardiomyocyte maturation after birth. So we didn't know what was RXR doing in cardiomyocytes. So what I first did was to generate a loss of function mice model targeting RXR's expression specifically in cardiomyocytes. We generated this embryonic double KO, and we started to see what was going on with them. And the first thing that we observed was, even though these RXR KO mice were born according to Mendelian ratio, not embryonic lethality, they died within 24 hours of life. This was very dramatic. The reason underlying this perinatal lethality was that indeed their hearts stopped working. Here you have a nice echocardiography acquisition. This is from day postnatal 0, the very same day the mice are born. You can see here how a control mice, their heart is nicely contracting. And this is how an RXR deficient mice is doing. You can see how their myocardium barely contract, indicated that indeed you need RXR expression in cardiomyocytes to allow cardiac beating and also neonatal survival right after birth. But what was the molecular mechanism that important for a cardiomyocyte that RXR was indeed controlling? So we were dealing with a transcription factor. So we did RNA-seq, and what we observed was that the major biological processes regulated by RXR were indeed related to metabolism, and more specifically, lipid metabolism. And here in this heat map, you can see how the genes involved in lipid consumption, lipid synthesis, and lipid homeostasis in general were completely blunted if RXR is not there. So, but what are the functional consequences for a cardiomyocyte if you cannot express the lipid oxidation genes right after birth? Well, that you cannot really produce lipid-derived ATP, indicating that indeed the reason of this lethal cardiac dysfunction we were observing was caused by an energy deficit coming from the fact that these hearts are unable to shift from the glucose oxidation profile to the lipid consumption one. So at that point, we knew, okay, RXR is a master transcriptional regulator. It is very much important for controlling this fetal to neonatal metabolic adaptation of the heart. But we knew that RXR cannot act alone. It needs to be activated by something else. So we were very intrigued by identifying what was the so important endogenous ligand for RXR in this context. And if I ask you to think about most important challenges that you face when you are born, you may think about maybe oxygen, But today I'm going to raise that one important physiological challenge that we all have faced at some point in our life is the very first time we ingest maternal milk. And turns out that maternal milk was indeed enriched in fatty acids, which have been proposed to be natural activators of these transcription factors. So we started to think that maybe maternal milk was indeed the potent physiological source carrying these RXR endogenous ligands right after birth. So in order to test this hypothesis, what we did was to nutritionally intervene the maternal milk composition in mice. We did that by feeding the dams by either a normal chow diet or a fat-free diet during pregnancy, aiming to generate a fat-deficient milk lacking the potential RXR ligands. We then fed control and RXR EDKO neonates with either normal milk or fat-deficient milk, and we investigated how this mitolipid gene signature, the signature that was controlled by RXR, was behaving. And as you can see here, the control hearts exposed to the regular milk having lipids were perfectly expressing the genes involved in this lipid oxidation and lipid homeostasis pathway. However, control mice expressing RXR, which were exposed to the fat-deficient milk, were resembling indeed the RXR deficient phenotype, indicating that fatty acids from maternal milk are indeed essential to activate this RXR-dependent transcriptional axis. And if you are not able to express the genes involved in lipid oxidation after birth, these mice also develop and resemble the RXR deficient phenotype, but undergoing lethal cardiac dysfunction within hours of life. So at that point, we knew, okay, the source of RXR ligands is the maternal milk. These are fatty acids, but which specific fatty acids? As you may wonder, maternal milk is enriched with many different ones. So we wanted to go further and particularly identify the nature of this fatty acid activating RXR. And to do so, we did lipidomics. And to make a long story short, we came up by identifying γ-linolenic acid, or GLA, as the natural endogenous ligand for RXR in our context. This GLA is an omega-6 fatty acid. This is an essential one, meaning that you are not able to synthesize it in the body. So it makes total sense that you need to ingest it at the beginning of the life. And we proved that RXR GLA was binding RXR by many different approaches, but here I want to show you the one that for me is the most relevant one, where we proved the physiological relevance of GLA in mice. Indeed, what we did was to rescue this fat-deficient milk phenotype by just restoring and again modulating the milk composition and just adding to this fat-deficient milk only GLA. And you can see here, when we add the GLA in the maternal milk, those mice that were dying within 2 days of life, approx, are able to thrive. And not only that, the presence of GLA in maternal milk, only GLA in maternal milk, is able to restore the expression of the genes that are controlling this lipid consumption pathway. We have shown with this work is a novel regulatory axis, GLA-RXR axis, that is critical for enabling the metabolic adaptation of cardiomyocytes right after birth. What we have shown is that in late embryogenesis, the RXR-dependent lipid consumption genes will be closed, inactive, their mitochondrial compartment immature, and glucose being the major energy source. However, once mice are born, RXR, which will be in the nucleus, will be activated by the GLA γ-linolenic acid coming from the maternal milk. And now that GLA-RXR axis is activated, they will be able to activate the expression of genes encoding enzymes located in the mitochondria that will allow this powerhouse to start oxidizing fatty acids, producing lipid-derived ATP, ensuring not only the extrauterine cardiac beating, but also the neonatal survival. So now we are proposing that this cardiac metabolic maturation, we can understand it as an intimate crosstalk between maternal and neonatal physiology. This work would have been possible with many people involved. So I would like to thank, of course, my PhD supervisor, Mercedes Ricote, and her whole team back in CENIC in Madrid, our close collaborator, Toño Enriquez and Raquel Justo, our collaborators in Sweden, Barcelona, Salamanca, Spain, and also Zenbio, and all our funding agencies. And of course, all of you for your attention, and I'm very happy to take any questions. Thank you very much.